Thanks, John. I think for this crowd, I'm actually going to skip over the intro. And I'm going to try to get through 3 topics. And so the first one is going to be more tutorial style about GWAS and the problem of hidden population structure, what can go wrong, and how you can solve that. And then I'm going to very, very briefly mention how you can leverage some of the insights and modeling from that problem to a slightly different but analogous problem for epigenetics. And that will be very brief. And then hopefully I have time to get into what is actually work in progress right now with Hilary Finucane on subphenotyping in GWAS. And what's kind of interesting about that, if you don't know what that means, I'll tell you when we get there, but it has a lot of the same flavor as these other problems in the sense that it's structure in the population that you're trying to uncover. So, right, I probably don't need to do this for this crowd. Everyone knows what a genome-wide association study is. And so probably many of you have already heard of this problem by now. And it's this problem of hidden structure. And so one of the fundamental assumptions in many statistical models is that the samples, or the subjects in this case in the cohort, are identically and independently distributed, or in statistical terms, abbreviation IID. But if you have a violation of that, things can go very wrong. And so some examples of violations include that you have people who are more or less distantly related to each other. So if you had pedigrees in the data, grandparents and parents and siblings, that would be one example. Or if you had different ethnicities, which is a much sort of broader structure, so people from different ethnic origins. And it could also be things like batch effects and stuff like that. So if you have this lack of IID-ness, and you ignore it, and you use a statistical model which assumes it to be true, then what you end up with, and this is now very widely known, are these spurious correlations, which sometimes people refer to as inflation of the test statistic. And so just to give you a little bit of intuition, this is a nice picture that David Balding drew many years ago in Nature Reviews Genetics. And it's sort of a very simplified version of the problem, but it's good to get it into your head. And so what it is it shows you a case control study. And inside of the cases and controls are these sort of hidden populations that you don't know about, let's say. And as many of you may know, that different populations or ethnicities have different distributions of alleles along the genome. And what's shown here is one particular position on the genome where there are A's and T's. And as you can see, if you look at population 2, there's more T's than A's. And in contrast, in population 1, there are more A's than T's. And so if you sort of did some sort of simple statistic using an IID assumption, and you just said overall in the cases, is the distribution of letters different than in the controls, then you would find indeed at this position there's sort of more T's in the controls than in the cases. And you might conclude that this very particular position was therefore correlated or causative of your trait of interest. However, if you were aware of these hidden subpopulations, then the more obvious explanation for this is actually what I just explained, is that these are sort of 2 ethnic subgroups which have different allele distributions. And because they're imbalanced between the cases and the controls, but you've ignored it, you end up with this sort of spurious association. And so, again, probably many of you have seen this kind of thing, but the way this has traditionally been diagnosed in the statistical genetics community is by looking at a sort of histogram or QQ plot of the test statistics from a large GWAS. So imagine you measure 1 million SNPs and you get a p-value for each one. Then under the assumption that this disease is not very polygenic, say that only maybe 100 SNPs or even 1,000, let's say, are significant, then you expect the remainder of the p-values, which is the majority of the p-values, to be drawn from the null distribution. And in this particular setting, the null distribution is simply uniform on 0, 1. And so you can actually just very readily plot what you expect the quantiles to be from this null distribution. That's on this axis. It's actually minus log p-values, so this is the very statistically significant region of the plot. And you can plot that against your observed test statistics. And this is real data here, and you can see this massive deviation from the null distribution, which tracks this diagonal and stays within the error bars. And so in this case, you know that either you have a tremendous amount of signal in your data, or that the assumption you've made that it's not very polygenic is incorrect, or you've actually just screwed up the modeling. And it's pretty easy to rule out this level of significance of p-values. It's actually a pretty small cohort. And so in this case, it's so egregious that you can pretty much conclude that you've done some mistake in modeling. And I won't go into it, but as data sets have become larger and larger and people can see polygenicity in the data set, it's been harder to tease apart what is this sort of confounding structure yielding inflated test statistics versus actual real signal. But I'm going to skip over that today. And so in contrast, if you take a model that actually is now aware of this lack of IID-ness and you kind of plug it in, then for this particular phenotype, you see it goes back into what you expect as a null distribution. And so, of course, what you generally hope to see is this kind of tracking the null distribution with a few outliers, which would be your signal and your SNPs of interest. So I mentioned that the problem here is this sort of lack of even similarity between people in the data set. And so it may not be surprising to know that any sort of modern method that tackles this problem uses at its core the pairwise similarity between all people. And so I've shown you this, and the pairwise similarity based on the genetics is normally what goes on. And so I'm not going to go into the technical variations of this, such as IBD, IBS. I'd say most people nowadays are using what is called, in some circles, the realized relationship matrix, which is essentially just a covariance matrix. And so I don't know if you can see in this particular light, but in this slide, there's no structure. So the off-diagonal is sort of uniform because every two people are equally similar or dissimilar. And the diagonal is just each person self-similar. And so this is an example where people are IID. And in the middle, you see this very fine-grained structure. This arises from pedigrees and things like this. And on the right-hand side, you have 3 blocks, which are each 3 different ethnicities. And within each block, it actually looks like this family structure. So this is a real data set. It's kind of an older one and almost a little bit contrived, but it's interesting to look at for these kinds of problems. And so I would say the current state of the art, which I'm going to work towards in this talk, just to at least give you some of the intuition on it, are originally Eigenstrat came along, which is based on principal components analysis, and now linear mixed models are kind of the state of the art, as well as combinations of these two. So I'm just going to walk you through this a little bit. So imagine that you actually don't know about this problem and you just want to use a regular statistical model which assumes people are IID. Imagine you have a quantitative trait that has roughly Gaussian residuals, then you can use linear regression. So what you do is you compute the likelihood of this model with a particular SNP in it, and then you remove the SNP and you can apply your favorite statistical test to get a p-value. So if you do that on this real data, which egregiously violates these assumptions I've discussed, then you get this massive deviation, as I've already shown you. And then if you do something a bit more savvy, which is this principal components or Eigenstrat, what you do there is you take this genetic similarity matrix, and as PCA is used in many fields, not just genetics. It's amazing actually how powerful it is. And so what it does here is essentially looks for the major axes of variation in this very high dimensional SNP space. So if the people in this room were the cohort and each one of you basically has like a million long vector of your million SNPs, and so you all lie in this million dimensional space, then what PCA does is it first looks for the single direction in that space that accounts for the most variation. And then to get the second PC, it looks for the one that's orthogonal to that counts for the second most amount of variation. And if you do this PCA, then what you can do is you're basically representing this very high dimensional space on a low dimensional manifold, and you can project each person. So each person in this room, you project them down on this very low dimensional space. Let's say it's 3-dimensional. So now instead of your million SNPs, you get 3 numbers, which tell you where you lie on each of these 3 axes. And then you add these as covariates to the model. And if you do that, you can do a pretty good job. And this, I love this plot. This is actually John's plot. And whenever I give a talk anywhere, people actually often email me for a copy of this, especially people not in genetics. And what it shows is if you take this matrix, this genetic similarity matrix, and you do PCA and you plot 2 of the axes, you can essentially recapitulate the map of Europe. And so it's not surprising that if the main problem in your data set is this sort of ethnic structure or geographic background, then this captures it well, and therefore it will help to correct the problem. So that's pretty good for capturing very broad structure, but if you have this more fine-grained structure, then simply by construction of principal components, you're truncating all the fine-grained signal because you're keeping only the top few principal components. And so that's where mixed models come in. And these actually go way back in the animal and plant breeding community, and were only more recently brought into human genetics. And so depending on your background, whether you're a machine learning person or a stat gen person or a statistician, you probably look at these models very differently. And so I actually like the interpretation of just this equation here. This is just the actual model likelihood. And what's nice about it is if I cross out this term here, this is the genetic similarity matrix, then what I'm left with is actually just linear regression written in vector form and nothing more. And so we know that the problem in linear regression for these kinds of data is that the samples are not IID, and that IID assumption is what is encoded here in linear regression. And so you can view this extra term in the linear mixed model as simply a correction of this IID-ness, and in particular, it's a correction which specifically tells you the pairwise similarity between every two people, which I've just told you is the problem. And so what I'm glossing over a little bit here is like, well, how exactly should you compute this? And I'll get into that a little bit here. So on this, again, this slightly contrived data, if you apply these kinds of methods, what you see is the naive model gives you this massive inflation of the test statistic. The red one is PC-based, and so it gets rid of the sort of three ethnicities. And then the last one is the mixed model, which takes you right into the middle. And so there's been a lot of research in mixed models, including by Eleazar Eskin's group here and many other people. And so my colleagues and I, Christoph and David, worked on this kind of area for quite a few years. And the kinds of problems that people work on in this space include that these models are very, very expensive. So I'm going to give you a sense of that in just a minute, but they scale cubically in time complexity with the number of people in the cohort. And so that can actually be extremely challenging and a real bottleneck. And then as people got into looking at how to make things more efficient, they started to also question, like, well, actually, what is the gold standard for these models if we could afford to compute anything? And sometimes there's an interplay between these as well. And so I'm going to give you a little bit of a flavor of some of the work we've done in this area. But other things you can also do with these models include testing sets of SNPs and testing richer phenotypes. So I'm currently working on longitudinal phenotypes. So why are these models expensive? So this is a multivariate Gaussian likelihood, and if you write it out, you don't really need to know what everything is here, but what you see is this K here is that genetic similarity matrix, and you see inverses of essentially K, and you see determinants of K, and these algebraic computations are the ones that kill you. These are the cubic computations. And so people have been trying to make these things faster for a little while. And we did two kind of speed ups in this space. One was exact, and one was an approximation. And so let me just tell you a little bit about that. So the first one, and they have some nice intuition, even if you don't sort of look at all the math in detail. But what you can think of is this is the likelihood of the mixed model. Again, I told you that if you just cross out this K, this is actually just linear regression written in vector form. And K, remember, is always this genetic similarity matrix. And what you can do is you can take the spectral decomposition of U, and what that essentially yields is you can think of U as like a rotation matrix. It's an orthonormal basis. S is a diagonal matrix. And what you can do is you can literally exactly rewrite the likelihood as follows, where you rotate the phenotype vector, you rotate your covariates, including the SNP you're testing, and what you're left with as a consequence of that is a diagonal covariance. So this is no longer linear regression because of this S term, but what's important is it is a diagonal matrix. So by construction, a spectral decomposition yields a diagonal matrix there. This is diagonal and this is diagonal. And what that means is that even though this is not linear regression, you can do computations in it with the same time complexity as linear regression. Because when you need to invert or take the determinant of a diagonal matrix, this is, it's pretty much free. It's linear in the diagonal components. But we still need to compute this thing here, and this is the bottleneck. That is still a cubic computation. There's no magic going on. And so something that we started to think about is, well, do we really need all of the SNPs to estimate this genetic similarity matrix? And so the standard in the field has been to take all available SNPs in your data set and then estimate this K. And so we started to say, well, we know, and I'll go over this in a second, yeah, there. So what we said is, well, we know due to the way the algebra works is if we can reduce the number of SNPs that go into computation of this matrix, then we can actually make things potentially much, much faster. And in particular, we can make it linear in the number of people instead of cubic. And so, and the reason for that is, I've told you now the main bottleneck computation is this here. This is the spectral decomposition. And this is the spectral decomposition visually. And what's important to know is if you take the spectral decomposition of this matrix, in general, it's cubic in n. But because it's an inner product or covariance or RRM matrix, it actually looks like this inner product of the SNPs, which have been normalized and unit varianced with itself. And this actually becomes linear in n and quadratic in the number of SNPs if you use fewer SNPs than there are people. And so now the question is, like, can you do that? Is that an approximation? Is that a good thing? What we do know is if you can do that, it really, really speeds things up, the time complexity and even the space complexity, because you don't have to store this whole vector. You actually never, this matrix, you never actually have to construct this explicitly. You can actually just use this and take a spectral decomposition. And so, right, so what we thought about at first in this was just that SNPs are somewhat redundant because of linkage disequilibrium along the chromosomes. And so we said, let's just thin out the SNPs. And maybe it will cost us something, but at least we get this. Now we can actually compute on data we couldn't formally compute on. And just to point out also, this was not some sort of theoretical computational bottleneck. There was actually data sets already several years ago where people literally couldn't use these models because of this problem. They would explode either in memory or you would have to wait forever. And so this actually worked pretty well. And then we started to think a bit more about what this model was. And again, like sometimes you can get to an intuition in different ways, but for us, the way we ended up with the intuition I'm going to tell you about is that we noticed in the literature, and we also knew from the machine learning community, that there's sort of an equivalence between the linear mixed model and Bayesian linear regression. So I don't know how familiar people are in the audience with Bayesian models, but Bayesian models in some sense you can think of as regular models where you put a prior on your parameter. And in this case, this is the prior here, alpha, for the weights of W, and W now is actually this W. So W are literally the SNPs that you compute genetic similarity with. And what I'm telling you is this is kind of like a regular linear regression. If I just take this is literally linear regression with my regular covariates, plus all those SNPs that I compute genetic similarity with, and then I just Bayesian with respect to those, and I integrate out their weights. And so why am I bothering to tell you this? Well, when you think about the model this way, what you see is that you actually have, thousands, if not a million SNPs here. And in general, you don't have that many people in your cohort. And if you're a statistics person or a machine learning person, you would basically never just throw in a million covariates just like that. You would do some sort of feature selection. And so that's what we started to think was we should be doing feature selection on these, and we don't actually need all of them. And that maybe is better than simply thinning the SNPs out along the genome. And if you look at it that way, some other implications arise from that. But the first is just sort of to recapitulate that is if you think of it in that way, then the right thing to do is to only put in SNPs which sort of carry some phenotypic signal. And that could be causal signal. It could be signal that represents this sort of ethnicity or family relatedness structure. And the other thing that was not known in this community, or people were sort of ignoring it, is that you need to be very careful not to condition on the SNPs you're testing. And so by that, what I mean is here, again, just focus on this equation. If you're testing a particular SNP here, and you also use it to estimate genetic similarity, then you're kind of shooting yourself in the foot, because you're controlling for the very SNP that you're testing, right? And because we integrate it out, the amount you shoot yourself in the foot is actually decreased, because integrating out is sort of like regularizing or shrinking the parameters. If you actually put it in as a fixed effect, you would literally never, ever get any p-values. And the other thing you need to be careful of is not only do you need to remove that SNP from the matrix, but you also need to remove SNPs that are very close by due to proximity on the chromosome leading to linkage disequilibrium. And so we started to essentially think about how we could remove the SNP and chunks of the SNP. And actually the sort of standard in the field now is that that's actually quite computationally burdensome. You can do some nice algebraic tricks to make it substantially faster, but it's still slow. And so what people now do is they do leave one out chromosome. So you basically compute a different genetic similarity matrix where in each case you leave out an entire chromosome. And when you test the SNPs for that chromosome, then you use that one. And so you have to sort of do 23 different genetic similarity matrices. Right. And so now, the question is also, I mentioned this sort of feature selection. I think I'm not going to go into the details of that. It's actually a very simple univariate feature selection. But what I do want to say is what was kind of neat after we did this is a paper emerged right around the same time from Oxford, where they basically, in Nature Genetics, it was a little bit, it's all simulation, so it's not clear this happens in real life, but one can imagine that it does. And they showed in their paper that under a particular type of this confounding factors, which roughly speaking had to do, imagine you have a little factory on a geographic grid which is polluting. And so it's a sharp, what they call sharp, because it's a very tiny, non-genetic, geographic-based risk, which acts as a confounder. If you have that kind of confounding, and you have rare variants, which of course are now sort of all over the place with next-gen sequencing, and you apply any of the known models, then you fail to control the type 1 error. And so you can see that here. All of these are inflated. This is actually null-only data. So by construction, it should be along this diagonal. And so what was interesting when we read this is we thought, well, if we apply our feature selection, we can probably actually solve this problem. And indeed, when you do that, you completely fix the type 1 error, and you still maintain some sort of power. And so, right, so this has just been a little bit of a flavor of some of the problems and challenges in this area, but actually beyond that, so there's been a lot of work in this. And so from our most recent paper, this is actually a table which now shows you recommendations depending on your data. So far what I've told you is that PCs can be a little bit limiting, and I've told you should do feature selection. But as it turns out, if you actually do a broader set of experiments, people, ourselves, in this paper we talk about this, and actually Tucker et al. In 2013 both discovered if you do this feature selection, and you only do this feature selection, you can actually sometimes still fail to control the type 1 error. And so there are different solutions to that. And there are different reasons for that, which I don't have time to go into here. But 2 ways to get around that is one, to do this feature selection for the limb, but also to include PCs. And I would say in general, you can actually get away with this. And the only time you need to go sort of whole hog and have this very expensive model, which is a limb where you have this one genetic similarity matrix, which is feature selected, and then you additionally have one that includes all of the SNPs. That will ensure you still control the type 1 error. And you can, if you put both of those in, you're pretty much guaranteed to be good to go, but it's very expensive. And I think in general, this is actually overkill. You would have to have a quite strong family structure to need that. But I just wanted to make sure everybody knew that that's sort of the current viewpoint, is that that just doing feature selection by itself is not useful. However, when you mix it with these other things that ensure you control the type 1 error, you still see increased power as a result of doing that feature selection. So now I'm just going to very briefly mention in a few slides a related project. So James was an intern with me actually here in LA when I used to be here. Yeah, that's right. And he said, I wonder if we can take some of these insights from this modeling and this problem in GWAS to EWAS. And so by EWAS, I mean epigenome-wide association. So the thing that, and I guess I don't need to, the epigenome are all these different kinds of things going on outside of the genome. And actually our paper focused on methylation along the genome. And so, but what's important for this talk is to know that this is cell type specific. And everything I've told you so far is genomic DNA, which is not cell type specific. So no matter where you got the sample from, it was sort of coherent, homogeneous, and it didn't matter. But now imagine you have some tumor, maybe breast cancer. This is a whole bunch of different cells, right? And now imagine what you want to do is you want to scan through the genome and you want to see at each site, is the methylation pattern at that particular site correlated or ideally causally implicated in the disease, just as you do for GWAS. But now you have different sets of cells, and each cell has sort of a different methylation profile. And so if you just naively, again, do this kind of simple analysis that assumes IIDness, you get actually something that's much, much worse than you ever see in GWAS. You get this huge deviation of the test statistic. And when I first started to give this talk, actually people used to come up to me and say, oh my God, this is what my data looks like. Please help me. And so at the time, the only way to fix this problem, which had already been identified, was through this sort of reference profile type of approach. And so what you would do, and this only worked in blood, and I'm actually not sort of currently working on this. I think that the only thing you can still do for these reference is based on blood. I don't know if people have been doing other ones. But what you do is you essentially hypothesize what the major cell types are, say in blood or whatever tissue of interest, and then you need to somehow purify and get just those cell types by themselves. And then you create a methylation profile for each of them. So now you know there's this sort of template for B cells, they look like this, T cells look like this, and so on. And given these profiles, you can do some reasonably easy modeling to infer the cell type composition for every sample. So imagine again that these people in this room are the cohort, and each person has its own difference in composition of cell types. You can kind of say, okay, I know that like for me, my sample, I was 20% B cell, 30% T cell, and this kind of thing. And then what you do, just as like an eigenstrat, is you include these as covariates. And if you do that, again, analogously to GWAS, you go from this kind of crazy looking plot where everything is tremendously significant to something where only a few things are significant. Right. And so what are the problems with this? So first of all, you need to get these reference cell profiles, and that may actually perturb your data, this process of purification. And much to my surprise, I didn't know this until a few years ago, that there's often you don't even know what the cell types are in many tissues. And it's not clear how well this generalizes from sort of lab to lab. And so we took as a sort of ground truth example this paper in Nature Biotech where they'd actually done cell counting, and so we knew some sort of ground truth. And you can see here just that this demonstrates actually between case and control there is sort of systematically a difference in the tissue composition. And in fact, because of that, what's a bit different here than in GWAS is that the p-values you get are actually, they're not confounding, they're not spurious, because here actually every methylation site is in fact implicated by virtue of the cell type composition differences between case and control. However, it's not interesting to say that everything is significant. And so you can kind of revise your goal and say, I want to know what's significant above and beyond cell type composition. And so what we did is we used these ideas from our FastLim work, which involved linear mixed models, and then we also added in principal components to do this in a reference-free way. And so just intuitively, you can imagine that this sort of cell type populations are somehow analogous to the ethnicity subgroups, and therefore, that this kind of might work. This is very empirically based. But when you do this, what's pretty astonishing is that you basically recapitulate on this data set what people get by actually including the cell counts as covariates. And there's only 5 methylation sites that are significant, and you get exactly the same 5 with almost identical weight estimates of them. And what's interesting also is in contrast to GWAS, if you ignore this problem, so this is uncorrected, and this is E-washer, which looks very much like the bronze standard, then what you see is these 5 significant methylation sites, they're basically buried below hundreds of thousands of other sites, and you would never see them. And in GWAS, it's actually not quite like this. After you correct, you actually often get a fairly monotonic transformation, meaning the ordering of your p-values doesn't necessarily change a lot, but the threshold might change. So we did this a couple of years ago now, and now Aran and Elior and these guys have actually looked in more detail at this, actually a bunch of people in this room, I think all of these guys. And so what they showed is actually that sparse PCA can actually do a better job in some settings. And actually Aran and I and Elior have been talking a little bit about maybe combining sparse PCA and the linear mixed model to do better still. So this is still an ongoing body of work in general. Right, so in my last, I have till 10, yeah. Okay, so in the last bit of the talk, I want to talk about, this is now actually very much work in progress. We're kind of halfway through, and then actually we had to take a break from this for various reasons. So Hillary was an intern with me now, I think last year? Yeah, last year. And we're going to start up again on this in August. And I'll just tell you where we are and where we're going with this problem. I think this is kind of I don't think we made up this term, the subphenotyping in genetics, but just to be clear about what we mean by that, is we mean that sometimes you have these labels 0, 1, so case control data, but actually embedded in there are these hidden distinct subphenotypes. And so people have been sort of positing that these exist for some time, and there's some diseases where it's actually really, people feel quite passionately that this is happening. So an example are in psychiatric diseases, and actually Jonathan Flint, who recently moved to Oxford, has looked a little bit at this, moved from Oxford to here, good catch, right. And so what is the problem? If you have 0, 1 data, but you actually have these distinct underlying phenotypes that have different genetic mechanism, and you treat them all as the same thing, then you're going to essentially deplete your genetic signal, whatever analysis you're interested in. So if you're doing GWAS, you're going to decrease your power. You're going to obscure your ability to understand the disease because you don't really know the causal SNPs, and you'll also decrease your apparent heritability. What's kind of, right, and so there are some examples, as I mentioned, so some subtypes are already known. IBD can be divided into Crohn's and ulcerative colitis. And also people are now using electronic medical records and seeing subtypes in autism. And type 2 diabetes is increasingly emerging as a set of diseases. One example here, an older one, is BMI. If you have high versus low, you have 2 different kinds of diabetes. So the neat thing, and like why is this in this talk, is that you can think of these subphenotypes as very analogous to population structure again. And it's a little different, right? So in the GWAS setting, we want to know what the structure is in order to correct for it because it's messing up our analysis. And here it's also kind of messing up our analysis, but we actually want to know what it is and it has more meaning. We're very interested in the finer details of it, right? And so, but what is interesting is there are a number of tools for studying population structure that can also be leveraged into this problem. And so we've been tackling this problem from 2 different angles, and I'll tell you a little bit about each of those. But first, let me tell you about how people in PopGen have taken random matrix theory to try to help solve or understand just looking for different subpopulations without any disease. Imagine you just have some group of people, like the people in this room, and there's actually 2 different groups, 2 different ethnicities. Can you use random matrix theory to help you tease those apart? And so what has been shown is if you take this genetic similarity matrix, here it's denoted as XX over M. It's again this covariance matrix divided by the number of SNPs, that's all it is. And you take an eigen decomposition of this, and you get the first eigenvector. And so that would be, where did it go? U1, so the top eigenvector of this matrix. And then you take V1 as the true class label. So imagine the right half here is one population, left half is the other population, 0, 1. And now you take the correlation between the true labels and the eigen vector, then this correlation converges to alpha. So now if alpha was 1, that would mean that the first eigenvector is perfectly correlated with the true labels. And that would mean that if you could just compute this first eigenvector, you would know precisely who is in what population, right? And now the question is like, when is alpha anywhere near 1? When is it above 0? And so what people have shown is that it goes through a phase transition. And the phase transitions, meaning it goes, for some settings, this is exactly 0, which means you can never see anything in this eigenvector. And in another setting, it's non-zero, and it increases with a certain set of variables. And so in particular, it increases the more people you have, that's n. So the higher n is, the easier the problem is. It also increases with the number of SNPs. And so the higher the number of SNPs you have, the sort of, assuming they're all tagging this, this difference, then the easier the problem is. And then also here is the fixation index, which describes how sort of divergent the 2 populations are. So if those 2 populations are actually identical and this is 0, then you actually revert to here and you can never capture anything. The larger this is, the easier the problem is. So that's all pretty intuitive, even if it's backed by this kind of bizarre random matrix theory. And so what Hillary did is she said, well, this is very interesting. Can we leverage this kind of analysis to get some intuition as to whether we could do this for subphenotyping? And so you can kind of use an analogous derivation, and what you do is you now say, imagine you took just the cases in this example of the subphenotyping problem, which are actually 2 different cases. Would you see a similar phase transition, and can you cast it in terms of variables that have more meaning for this problem? And indeed you can. So again, when you take the first eigenvector of this very same matrix, which now is just the cases. So this is actually a bit stupid because this is a case control study and we're ignoring the controls. And then you get this phase transition, and this is the quantity which you need to increase. And so let's break down that quantity a little bit. So what it says is, remember, the larger tau is, the easier it is for me to use PCA to identify the subphenotypes. And what this is the genetic correlation between the two traits. So imagine that the two traits are actually identical, which means I don't have subphenotypes. Then this is equal to 1. This collapses to 0, and the problem is impossible. So that makes sense. This here says that the larger the heritability, the easier the problem is. And that's also not surprising. That just means there's more signal in the data. And this is interesting, this says that the fewer the number of SNPs, the easier the problem is. And what that actually means is sort of, there's a trade-off between you want to identify the SNPs that kind of convey all the heritability. And to the extent that there are few of them and you can get it down to a few, the problem becomes easier. And so this leads to ideas about how you also might want to use feature selection. And I think I'm going to skip over this one. It's less intuitive. But so you can do some simulations and you can see this phase transition as you change this quantity. And I should say what's nice about this is, this is a little bit sort of too idealized and conceptual. And also I should say that this assumes right now the heritability is the same between each of the subphenotypes. You can generalize this, but nevertheless, this isn't sort of exactly where we want to end up. But what's nice about it is it gives you a sense for a given data set. If you know your sample size, if you can take a guess at the heritability and maybe sort of bound the shared genetic correlation and know the prevalence, like you can get some idea of like, do I have any hope of actually tackling this problem on this data set? And so I think that's nice. A lot of people have tried to tackle this problem in the past that we know, and actually none of them have had any success. So we haven't yet got to real data, but hopefully we do. But the problem with PCA is like this is kind of neat for helping to understand these parameters and how hard the problem gets as you change them. But we kind of want a more mechanistic model that lets us actually kind of do a GWAS, if you will, under this setting where we don't know what the subphenotypes are, but it is sort of this case control. And so you can use just a latent variable multinomial logistic regression. So what that means is, remember, logistic regression is for 0, 1. And if you were in a GWAS setting with no confounding population structure and these kinds of things, you could just use logistic regression for case control. And so what we're going to do now is we're going to say it's not just 0, 1, but the ones are actually broken down into, say, 3 different subphenotypes. We'll have to specify that ahead of time and then do model comparison. And then we're going to learn those as hidden labels in the data. And so it sounds all very easy. Of course, this is actually very hard to do statistically. So this is the direction we're going. But what's nice about this approach over the PCA approach is, one, we get to now include the controls in there, which is not throwing out data, and a bunch of other things I'll mention actually on the next slide. So, and you can do simulations with this, again, changing these, and you see actually basically comparable ability to tease apart the populations based on this latent logistic regression model as compared to PCA. But the latent logistic regression model is nicer because it's more interpretable and more generalizable. So that's where we are now, and some of the ongoing directions are we're going to do more simulations, but obviously the idea is to look at real data, and you really do need large sample sizes here. So we've paid several £1,000s to the UK Biobank to look at some phenotypes there we haven't had a chance to do. And what's really hard about this problem is it's totally non-convex. There's lots of local optima, and so even if you had the signal there and you could get it, it's actually, you need very smart initializations. And so one of the ideas we have is to use this sort of PCA eigenvector as one kind of initialization. And then people always say, what are you going to do if there's confounding structure? And so for starters, we have ignored that because that's really hard. Imagine you have both these subphenotypes and you have confounding, and you find these groups. Like, is the group actually related to the disease, or is it just these sort of ethnicity-based subgroups? But what's nice is, under some assumptions, like imagine that you assume this confounding factor is the same in the cases as in the controls, then you know the controls don't have any subphenotypes. And so you can, in a way, hopefully correct for it in that manner. And then I think using only the genetics for this is going to be actually very, very hard. And so we want to do some maybe smart regularization using pathways or genes to make it a more sort of statistically efficient. And also probably use auxiliary data, so clinical or molecular data like gene expression. But this is very much ongoing work. And now just a small pitch. So I'm in Cambridge, Mass now, and we actually once a year or two we hire a postdoc in comp bio. So there's going to be an opening 1 year from now for that. And I don't think this is appropriate to this crowd, but if you know anyone who's a talented undergrad and just wants a year gap year to get some research experience, we also hire people, and we also have a PhD intern program. Thanks.